Hi, I'm Xavier, and this is my science fair experiment, the effect of manure from cows, horses, and chickens on the activity of methanogens. So some basic background information before we get into my experiment. Basically, there are these things called anaerobic digesters. You put manure in them, and the microbes in the manure break the manure down into methane. Those microbes are called methanogens. And that methane can be captured and burned as a relatively clean fuel source since it's natural gas. The purpose of my experiment is to find which type of manure, cow, horse, or chicken, produces the most methane so I can maximize the efficiency of anaerobic digesters. So what you need to know about the digestive tracts of the different animals I'm working with: cows are ruminants, so basically they have a rumen, which is like a really large stomach that allows for food that they eat to ferment for a long time and grows a lot of methanogens. Horses and chickens just have a cecum, which is this tube-like hindgut that allows food to pass through very quickly and doesn't cultivate as many methanogens as a rumen does. So because of this, I can create my hypothesis, which is if cow manure is placed in an anaerobic environment, then it will produce more methane than chicken manure or horse manure in the same environment because the cow digestive tract fosters more methanogens than the digestive tracts of horses or chickens. Wow. And there is a picture of an anaerobic digester to give you a little bit of scale and an idea of what they look like. So now onto my procedure. I put manure along with sand and water to create an anaerobic environment for that manure in a mason jar, and I did that 4 times for each manure: cow, chicken, horse. I then autoclaved some of the mason jars, which killed all the microbes in them to act as a control group. I then let them sit in an incubator for 5 days and took gas samples from them and put them in a gas chromatograph after those 5 days to see how much methane they made. So on the left you can see my setup, that blue box looking thing. On the very left of the left picture is a gas chromatograph, and then to the right of the gas chromatograph, plugged into it, is my computer, which is recording all the data and making a graph, and then you can see my mason jar manure samples. The right picture is how I gathered my gas samples. Basically, I wrapped parafilm, which is like this cellophane wrap, around the lids and put a syringe into the mason jar so that not a bunch of gases escaped. This is the typical graph from most of my manure samples, and basically there was one peak for air, which is in the start, and one peak for methane, which is the next one. Now the height of the peak for methane is it differs between the manures, and that's where I get into analyzing my data. So after I cut out outliers, I made this graph, which is, it shows that chicken manure produced a lot more methane than cow or horse. Yeah, that was a surprise to me. So how did I cut out my outliers? Well, I used a 1.5 IQR test to remove outliers from each livestock type, like cow, horse, chicken, and then I used a one-way ANOVA test to compare the retention times from the different samples to a confirmed methane sample to make sure that it was methane I was looking at. And in that test, I found out that all of my control groups, all of my control samples didn't have methane in them. So, yeah. My conclusions: I finally used a one-way ANOVA test and a Tukey-Kramer test to find out that chicken was significantly different than cow or horse, which were pretty much the same. So chicken was the clear winner. It made the most methane, and that is my project. Thank you guys for listening.